After studying this module, you will be able to know about electrophoresis, about matrices supporting protein electrophoresis according to molecular weight, about page, STS page and other types of page. Application of electric current across is a mixture to move charged molecules in solution is known as electrophoresis. This technique has extensively developed for molecular separation as in presence of electric charge, molecule moves with a speed depending on their shape, charge and size. It is a rapid and simple technique. Purification and analysis of very large molecules such as proteins and nucleic acids is mainly carried out using electrophoresis. It can also be applied to simple charged molecules which includes amino acids, sugars, nucleotides, peptides and simple ions. To monitor and analyze electrophoretic separations, highly sensitive detection methods have been developed. Thin layer of sample is applied to a solution stabilized by a porous matrix to carry out electrophoresis of macromolecules. The molecules move at different velocities through the matrix on application of voltage. After completion of separation, various species are detected as bands on different positions in the matrix. Electric current when passed in the solution produces heat which can cause diffusion and mixing of bands. To prevent mixing of these bands, matrix is used. It can be made of various number of materials such as cellulose, acetate, paper or gels made of agarose, starch or polyacrylamide. The matrix acts as size selective sieve in the separation in case of agarose gels and acrylamide. Staining or autoradiography of the separated molecules is done at the end of the run to detect its position. For quantification, densitometer is used and for storing it permanently, the gel can be dried and stored. Most commonly used stabilizing media is polyacrylamide and agarose gel. These gels are formed as thin, flat sheets or slabs or as cylinders in tubes. The most common matrix used for separation of protein is polyacrylamide. Depending upon the size of molecule to be analyzed, nucleic acid is separated on agarose gels or polyacrylamide gels. While setting up an electrophoresis unit, the gel is mounted between two buffer chambers in a particular manner in order to make sure that the only electric connection between the two chambers is through the gel. Contact between the buffer and gel may be direct liquid contact or through a wick or pad of paper or gel material. Depending upon the separation, distance needed and the amount of sample gels can be of all sizes. Matrices supporting protein electrophoresis. Electrophoresis is carried out in a matrix which serves the purpose of separation of different size of molecules. Smaller electrophoretic mobility is seen in molecules of large size. Basically, two types of matrices are used for protein profiling in forensics. These are papers such as cellulose acetate or gels like agarose or polyacrylamide. Separation by molecular weight. Electrophoresis is widely used to separate proteins on the basis of their molecular weights. Functions of proteins can be determined by using native or non-denaturing electrophoresis. But sometimes some proteins are not separated by this electrophoresis and required denaturing of proteins. Separation after denaturing is known as denaturing protein electrophoresis. Properties of matrices supporting protein electrophoresis by molecular weight. When agarose is used as supporting matrix, it has larger pore size, fragile strength, mainly used for nucleic acid, and is non-toxic. Whereas polyacrylamide has smaller pore sizes, high strength, it is mainly used for proteins, and it is toxic. Agarose gels. For separation of large protein, agarose gels is used. Agarose is a derivative of agar and is highly purified polysaccharide. It is normally purchased as dry powder. Heating the suspended powder near boiling helps it to dissolve, and it remains as liquid till 40 degrees centigrade when it gels or sets. Various other types of agarose are present which have lower gelling and melting temperature as compared to standard agarose. Recovery of material from gel is enhanced by such properties like separation. The concentration of the agarose gel determines the pore size and sieving characteristics of gel. The size of pore is small with higher concentration. These gels are fragile and should always be handled with care. These gels are held by hydrogen and hydrophobic bonds which make them brittle. Polyacrylamide gels. These gels are comparatively tougher as compared to agarose gels. This gel forms when a mixed solution of acrylamide and cross-linker monomer copolymerize into long chains that are covalently cross-linked. Cross-linker held gel structured together. This polymerization of acrylamide is a free radical catalyzed reaction. Preparing these gels is more complex process. Preparing and pouring the monomer solution. Atmospheric oxygen inhibits polymerization. So for constant results, the acrylamide monomer should be made free from air by exposing it to vacuum for a few minutes. To avoid oxygen problems, minimum stirring should be done. While pouring the solution in the mold, meniscus is observed on the top of the solution. Ignoring the meniscus can cause polymerization with curved top. This will lead to similar curved pattern in all the bands after separation. Before polymerization, a thin layer of water or water saturated N-butanol should be layered on the gel mixture to eliminate the meniscus. This water or butanol layer is poured off after polymerization and flat surface of gel is obtained. This layer of water or butanol also prevents oxygen, thus preventing the inhibition of polymerization on gel surface. Alternatively, a flat etched form such as a comb can be inserted into the top of the solution to give a mechanically flat surface. Care must be taken not to trap small air bubbles under the bottom edge of a comb. Combs made of Teflon will inhibit polymerization of a thin layer immediately next to the comb because of oxygen dissolved in the plastic of the comb. This unpolymerized layer is convenient because it eases removing the comb without affecting the performance of the gel. Determining the pore size. Two parameters determine the size of pores, that is total solid content %T and the ratio of cross-linker to acrylamide monomer %C. The ratio of sum of weights of acrylamide and cross-linker solution is known as %T. By varying the amount of cross-linker, size of the pore can be adjusted. The weight by weight percentage of total cross-linker weight in the sum of monomer and cross-linker weights is known as %C. In case the mixture to be analyzed has wide range of molecular weights, then pore gradient gel should be used. In such type of gels, the pore size is smaller at the base and larger at the top. The gel becomes more restrictive as the run progresses. The presence of the gradient yields a gel with a wider range of size, resolution, and also keeps bands tighter as compared to uniform concentration gels. Polyacrylamide gel electrophoresis. Page is used for separation of proteins. The acrylamide gel used serves as a size selective sieve during the process of separation. The mobility of smaller molecules is faster than the larger ones. In most of the page applications, the gel is mounted between two buffer chambers and the only electrical path is through the gel. The gel is kept in vertical orientation most of the time. Comb is used to generate wells in the gel in which samples are applied. Application of electric field across the buffer chambers forces the proteins to migrate through the gel. Two types of buffer systems can be used: continuous buffer system, discontinuous buffer systems. Continuous buffer systems, use of same buffer at constant pH in the gel, sample and electrode reservoirs. Samples are loaded into wells and proteins that are closer to the gel enter first. This provides a uniform separation matrix but yields fuzzy and unresolved protein bands. Continuous systems are rarely used for protein electrophoresis but commonly used for nucleic acid analysis. Discontinuous buffer systems use a gel separated into two sections, a large pore stacking gel on top of the small pore resolving gel, and a different buffers in the gel and electrode solutions. Proteins migrate quickly through the large pore stacking gel, and then after entering the small pore resolving gel, it is slowed down. Resolution is improved by stacking the proteins on top of one another, forming a tight band. Discontinuous buffer systems provide higher resolution than continuous systems, and varying the buffers used in the sample, gel and electrode chambers create a variety of discontinuous buffer systems that can be used for a variety of applications. Discontinuous native page. The original system was developed by Davis and Ornstein to separate serum proteins in a manner which can preserve native protein conformation, biological activity and subunit interactions. The buffer used to prepare proteins is non-reducing and non-denaturing. No reducing or denaturing agents are used for electrophoresis. Preservation of native charge takes place and lead to migration of proteins towards either electrode. This separation produces unpredictable separation patterns which are not suitable for molecular weight determination. Therefore, this technique separates proteins in their active state and can resolve proteins of same weight. STS polyacrylamide gel electrophoresis. Lamellae 1964 incorporated the use of detergent sodium dodecyl sulfate STS to overcome the limitations of native page systems. This led to creation of most popular form of protein electrophoresis, STS page. Complete denaturation of proteins takes place through use of detergent STS and other denaturing agents, and they dissociate from each other. Non-covalent bonding takes place between STS and proteins in a manner that imparts an overall negative charge on the proteins. Since STS is negatively charged, it masks the intrinsic charge of the protein it binds. A similar charge to mass ratio for all proteins in mixture, since STS binds at a consistent ratio of 1.4 grams STS per 1 gram protein STS, a stoichiometry of about 1 STS molecules per 2 amino acids. A long rod-like conformation on the proteins instead of a complex tertiary shape. So therefore, migration of STS coated protein totally depends on its size. According to the original technique developed by Lamelli, STS was introduced in the gel, but now incorporation of STS in gel is not required. STS in the sample buffer is sufficient to saturate proteins, and the STS in the cathode buffer maintains the STS saturation during electrophoresis. Other types of page: blue native page, BN page. This technique helps to separate and characterize large protein complexes in their active and native forms. BN page was originally developed by Scheger and von Jagow in 1987. Mild and neutral detergents are used for solubilization of protein complexes. Kumasi Brilliant Blue G250 is negatively charged stain which binds to the surface. This imparts a high charge to mass ratio that allows the protein complexes to migrate to the anode as they do in STS page. But denaturation and dissociation of protein complexes does not take place as compared to STS. Zymogram page. Detection and characterization of collagenases and other proteases within the gel is detected using this method. Casting of gel is done using gelatin or casein. This acts as substrate for the enzymes that are separated under non-reducing condition in the gel. For separation according to molecular weight to take place, proteins are run with denaturing STS. Enzymes are renatured and then are allowed to break down the substrate. After this process, the zymogram gels are stained with Kumasi Brilliant Blue R250 stain, which stains the substrate while leaving clear areas around active proteases. Summary: application of electric current across a mixture to move charged molecules in solution is known as electrophoresis. This technique has extensively developed for molecular separation, as in presence of electric charge, molecules move with a speed dependent on their shape, charge and size. It is a rapid and simple technique. To monitor and analyze electrophoretic separations, highly sensitive detection methods have been developed. The matrix acts as size selective sieve in the separation in case of agarose gels and acrylamide. Staining or autoradiography of the separated molecules is done at the end of the run to detect its position. Most commonly used stabilizing media are polyacrylamide and agarose gels. Depending upon the size of molecule to be analyzed, nucleic acid is separated on agarose gels or polyacrylamide gels. Similar electrophoretic mobility is seen in molecules of larger size. Basically, two types of matrices are used for protein profiling in forensics. These are papers such as cellulose acetate or gels like agarose or polyacrylamide. Some proteins are not separated by this electrophoresis and require denaturing of proteins. Separation after denaturing is known as denaturing protein electrophoresis. Agarose is a derivative of agar and is highly purified polysaccharide. The concentration of the agarose gel determines the pore size and sieving characteristics of the gel. Polymerized acrylamide gel forms when a mixed solution of acrylamide and a cross-linker monomer copolymerize into long chain that are covalently cross-linked. Atmospheric oxygen inhibits polymerization. Before polymerization, a thin layer of water or water saturated N-butanol should be layered on the gel mixture to eliminate the meniscus. Two parameters determine the size of pores, that is total solid content %T, and the ratio of cross-linker to acrylamide monomer %C. The acrylamide gel used serves as a size selective sieve during the process of separation. The mobility of smaller molecules is faster than the larger ones. Use the same buffer at constant pH in the gel, sample and electrode reservoirs. Use a gel separated into two sections, a large pore stacking gel on top of a small pore resolving gel, and different buffers in the gels and electrode solutions. The original system was developed by Davis and Ornstein to separate serum proteins in a manner which can preserve native protein conformation, biological activity and subunit interactions. Discontinuous native electrophoresis technique separate proteins in their active state and can resolve proteins of same weight. Lamelli in 1964 incorporated the use of detergent sodium dodecyl sulfate to overcome the limitations of native page systems. Complete denaturation of proteins takes place through use of detergent SDS and other denaturing agents, and they dissociate from each other. BN-Page technique helps to separate and characterize large protein complexes in their active and native forms. BN-Page was originally developed by Shager and von Jagow in 1987. Kumasi Brilliant Blue G250 is negatively charged stain which binds to the surface. Detection and characterization of collagenases and other proteases within the gel is detected using this method. Casting of gel is done using gelatin or casein. The Zymogen gels are stained with Kumasi Brilliant Blue R250 stains, which stains the substrate while leaving clear areas around active proteases.